Hey everyone, I'm Jeff Guyon, I'm from Charleston. So you want to save the ocean. I mean, if you're anything like me, you want to do something along the way. You want to make a difference. So I had this title, so you want to help save the ocean, let's do it. And Steve Moore helped me and added that other section that we have along the way. That application. Sure. So I'm in NCCOS, which is the National Centers of Coast Ocean Science, which is part of NOAA or NOS, National Ocean Service, part of NOAA. This is kind of where we get stuff. This is, the authority for what it is that I do. So NOAA's mission is to conserve and manage coastal and marine ecosystems and resources. So NOS is the National Ocean Service, provides data, tools, and services that support coastal economies and their contribution to the national economy. The last one, this is our organization. We deliver ecosystem science solutions to sustain thriving coastal communities and economies. So that's what we do. That gives us the authority to work on the ocean. So this is where I work. So it's a beautiful building. It's in Charleston, South Carolina. So it's called the Hollings Marine Lab. And this is what my branch does. We conduct health assessments in key marine species, producing actionable science. We want to do science that makes a difference. To help support the management of NOAA living, and you're going to see living resources like corals and dolphins, and physical trust resources like near sites and sanctuaries. So this is just some quick introduction slides, and hopefully get to some fun slides. I'll be back and forth. So this is me. This is my branch. We do very different things. We have a number of different programs that are in there. We have the Coral Health and Disease Program, led by Dr. Cheryl Woodley, with a number of contractors, really amazing contractors. So we work on shallow water corals. We have our Coastal Marine Mammal Assessment Program. We work on dolphins. Oh, yeah. Yeah, I don't know how this goes, but we'll hold it. Kind of fumbling here. So in the Coral Health and Disease Program, we've had one neurosurgeon, Victoria Salinas. Victoria here? Good to see you. We've done this all virtually, so I appreciate everybody coming on the way. So Victoria always amazed at the depths of water that she's diving in and the work that she's doing. So she's working with Cheryl's group. But just an amazing group. We'll go through a little bit of what the Coral Health and Disease Program does. We have a coastal marine mammal assessment program. We work on marine mammals, dolphins, baby sperm whales, and pilot whales, stuff like this. Led by Wayne McPhee, just an amazing person. As Marcus is doing an ergo. Marcus is doing an ergo with folks up in Sandy Hook, the Northeast Fisheries. You have our environmental genetics program, led by Dr. Thomas Gregg. And in there I put Margaret. I know Margaret's here. You work with this, and Margaret and I did it on Nerdo. Paul and Leah, is Leah here? Leah. Good to see you. So thank you for coming. And we have a microbiology section. So you want to say the ocean, there's got to be a problem to solve, right? But if you're going out there, there's something happening. You want to use the resources that you have to try to fix something, make the world better. So save the ocean problem. What I want to go through is some of the things like we've done in our group to try to make the world better. So in 2019, there were 4 pilot whales that stranded on Edisto Beach. So it's not necessarily unusual. We respond, actually it's not we respond, but we work with a stranding network that responds somewhere between 30 and 50 marine mammal strandings in South Carolina. But as you can point out here, so these are Tessa, she's a contractor in our group. This is Francesca, she worked for the Stranding Network. And these are 2 College of Charleston students. So I think I took this picture, got down there, it's a whole day thing. They pick up these mammals, they got euthanized, so they come across. They're also crowds. people care what's happening. Like, what's happening to those marine mammals? Why did they die? Why did they strain? So what's causing that? So our group will go out there and do a necropsy. You'll see a picture of it. Let's do a necropsy. I try not to pass out, but do a necropsy. So it's always in the hot sun, like 90 degrees outside when we're doing this. So in there, what we do is we want to understand what are the impacts on coastal communities. There really isn't a better organism to study like as a surrogate for human health than a marine mammal. So that's another reason to work on marine mammals. This is Wayne McPhee. And Wayne's doing a necropsy, and I hope it's right. It looks like a pygmy sperm whale to me, but I'm not positive. But Wayne does work on a number of different things, like what are the pathogens that are impacting marine mammal health? What are some of the entanglement issues with people with crab pod buoys out there that get entangled? Fishermen will have lines with their mouths problem. We'll have and microplastics is one of them. This is a picture of Wayne giving a talk in Spain. So I thought it was kind of neat. Besides it rhymes, Wayne. Something. If he caused enough trouble, they help. Okay, save the ocean problem 2. Anybody recognize this? Coral. So the shallow water coral. This is stony coral tissue loss disease. There, so in 2014, there was a disease outbreak that started near the port of Miami. It's kind of associated with at least in time-wise, temporarily with the dredging operation that happened about that time. The disease has spread north of Miami and gone down to the Florida Keys Reef Tract, down towards past the west and on the way to Dry Tortugas. So there's maybe 40 coral species that live in that Florida Coral Reef Tract. About 20 of them are susceptible, 20 species. It's not the only coral disease that's out there. There's a number of other ones. So black band disease, white band disease, and other diseases that are out there. So we have in there's a problem. So in there, sorry, it's hard to breathe through the mask. But in the of the 20 species that are susceptible to disease is almost completely lethal in a few. Some of them are extremely endangered, like the pillow corals I'm going to show you. And they're very susceptible to this disease. So we've gone ahead of that disease track, worked with us and a number of academic researchers and other government researchers, and collected samples of those species before they got impacted by the disease. So we have, that's our problem. We have a program that works on that. That's the Coral Health and Disease Program. It's led by Dr. Cheryl Woodley. And Dr. Cheryl Woodley edited this book. So I tell her she actually wrote the book on the stuff that we do. She corrects me and she goes, I didn't write it, I edited it. But I count it the same. So we have a facility there in Charleston. We grow anywhere between 8 to 10 species of corals that are both in the Atlantic and in the Pacific. We try to understand what's causing that. So this is some work that was done on sedimentation. So they have dredging operations that's stirring up sediment. What happens to the coral? Because now the corals, which are dependent on light getting through, can't get to the light because the sediment's blocking it, or it's covering it and filtering it. So it's causing different problems. And we have Cheryl's group, which has a number of different projects, and we work on those. Basically, all the different types of coral diseases, impacts to the Zooxanthellae and other. So where are the places that we work on stony coral? So we're located in Charleston, South Carolina. In Charleston, we don't have many stony corals. We're offshore, there's one species or so, it's in fairly deep water. But it's not like the Caribbean or Hawaii. So where does our work take us? It takes us to the Virgin Islands. We work with the National Park Service. We work, this is the sedimentation work that was just north of Miami. That's the Florida Keys, or the National Sanctuary that's in Florida. We work out in Hawaii for issues that have to do with contamination that's getting into Maui. Plus, you know some of the sunscreen work that's coming out. So sunscreens in Panama Bay. So that's connected to the work we did. So Cheryl published on that type of work. And we have work in the far Pacific, the Pakistani Bay, which is the American Samoa and African sector. Your international work, that's not good. So Save the Ocean problem 3. There's only 4, so don't worry, you're not going to go into this rest afternoon. You got 4. So problem 3. So people recognize this. So some Deepwater Horizon, some of the stuff that happened there. So in there's a group that went through in our group, not my group, but another group. There's issues at the top of the surface. Oil floats, and they have different spectrums and issues that went through it. So our groups work with things like marine mammals, impact of marine mammals that might have gone through. But what I want to highlight here is another group worked both in NCCOS in our organization. The study the impacts of oil from the spill, not at the surface, but at the very bottom. So there's deep water corals that exist way out there where the Deepwater Horizon spill was. Those corals were impacted. And so they had a project to try to restore some of those corals that were negatively impacted from the oil spill. So they've been out there on NOAA ships. I haven't been out there on NOAA ships, but others have been. And they collected some deep sea coral species that they have. And they want to know what species are they? Are they genetically the same, genetically different? How much variability exists there? So we have a genetics program that took this, isolated some DNA, and did genetic ontology work. They showed that these 2 species, so I'm pretty sure we're going through this here, but we're going to see a penicula, which is right here, and you have Hypnogorgia, which is right here, co-located. So what we thought were 2 different species, in this case, we're 1 species. So they look different, but they're the same. And so there's things that with genetics that you can find that are very difficult to tell elsewhere. So the last problem number 4. So can anybody see the problem? No, because it's blurry. So I'm going to get rid of the blurriness, okay? So now, can you see the problem? It's a trick question because this is an American Samoa. This is Pakistani Bay. I'm going to show you this a little bit. It turns out that there wasn't much of a problem at the time. It doesn't mean there's nothing wrong. But sometimes the work that we do is on things to understand, is there a problem? So this is in Pakistani Bay in American Samoa, the National Marine Sanctuary that's out there. Beautiful bay. And at the very top of there is one of the dump sites they have on American Samoa. There's a drainage stream that comes out and flows into the bay. They want to know, what are the impacts on those corals that are going around the bay? So they collected pore water and some of the microbiology work that was done there to understand if there's like fecal coliform bacteria and impacts to the life that's there. The results showed that the bay was pretty healthy. So everything was going, pretty good. You can't say it's perfect, but it was pretty healthy. So the question is, what are you concerned about? What drives you? What gets you up in the morning? So this is Dr. Cheryl Woodley, the one that wrote the book. This is part of her group. We have a coral facility that maintains a lot of different tanks of corals. So corals come from, again, I think we have maybe 8 to 10 different species from the Atlantic and Pacific. These are those pillar corals that, again, were decimated by stony coral tissue loss disease, collected ahead of the disease outbreak. They have specific genotypes, and this is the only place in the world they existed. In places, other labs, and other ones. But I think we shared them since then. Because if something goes bad here, then you lose them forever. But that's them. And they made it into this postcard for the field. So when I look for inspiration, I look at history, how did other people do things? So this is Admiral Grace Hopper. She has a PhD. She graduated from Vassar College in around 1930. Went to Yale, got a PhD in mathematics, joined the Navy at the head of World War II. went to Harvard, worked on their Mark, whatever it was, computer that they had, and wrote some of the initial compiling language that they had. The initial compilers were done by Grace Hopper. So from that, they have a ship named the Grace Hopper destroyer. There is at the Naval Academy, the Cyber Warfare Center is named after Grace Hopper. This is down here. There's a building at Yale named after Grace Hopper. What made her famous for me is that I rode in an elevator with once. It wasn't mentioned anywhere. But in there, like I was a computer scientist person, so I went to a bunch of lectures, and she gave the lectures and seminars. And she was somebody that I wanted to meet, somebody that was a hero of mine. So I got to my place where I lived in Washington, D.C. I get in the elevator, and she walked to be behind me in the elevator, and I couldn't believe it. So I'm like, oh, because I lived right across the Pentagon. And so when she got in, you push the buttons on the elevator, and it would light up with whatever floor you wanted to go. So I didn't touch anything because I wanted to see where she went to, you know. So she pushed 16, and I lived on 16. I'm like, wow. So she gets to the top, and it bifurcates into 3 different branches. So I wait there, and she goes in the same direction I did. And I'm like, she lives on the same hall. So I was really excited, and I went to work the next day, and my friends told me Grace Hopper lives there. And so I said, I got on the elevator with Grace Hopper, and I tell a lot of stories, you know. So they said, yeah, how do you know it's Grace Hopper? I'm like, she was in an admiral's uniform and had a name tag that said Hopper. So I'm confident it was Grace Hopper. So this is the last slide that I've had. Put in here, I took this from the USNovelist.org, an interview that was done by a person I don't know, Liz Gilbert, with then Captain Grace Hopper in 1981. But we'll read that. Grace Hopper believed that her greatest accomplishment was the training she gave to the younger generation. The most important thing I've accomplished, other than building a compiler, is training young people. They come to me, you know, and say, do you think we could do this? And I say, try it, and I back them up. I like that a lot because she doesn't say, I'm going to tell you how to do it. She doesn't say, like, this is what you're going to do. She works with people that have the inspiration, that want to do something. And they go out there, have their idea, and present it to her. And she supports that. So, and I think about that with the folks that I work with in my branch. But I also think about the students that we had, the neuro students we had, you know, work with Margaret, you know, we ran into problems. There's always problems, there's never a lack of problems, but we pivoted along the way and found a way to address them. And Leah, we did the same thing, you know, so there's times in there, and Leah's helped out with a lot of the programming that we need, and it worked out well. So we ran into problems, found a way. It wasn't me that came up with solutions, you guys did, and we worked around it. So anyways, I just want to leave you these words from Grace Hopper and the application and stuff, everybody. I want to thank you for giving me the opportunity to. This is the first time really I've gotten out, so I wear the mask and stuff as I go through. So it is really good to see.
All right, we got about 5 minutes to ask Dr. Guyon any questions.
Oh yeah, other famous people I've met.
Or yes.
Yeah.
Is NCCOS partnered with NOAA or section within NOAA?
So it's I'm a little confused all the different things called line offices and stuff like this. So the National Centers of Coastal Ocean Science is part of the National Ocean Service, which is a line office of NOAA. So it's part of NOAA. Before this, I worked with the National Marine Fisheries Service. So I tend to move around some. I think at least for me, I tend to find a challenge and work with folks, and then I like to learn new things. And so before this, I worked in Alaska. So I managed the genetics program for the Alaska Fisheries Science Center there. And I did a rotation as the lab director of the Galveston Lab in Galveston, Texas, which is really nice. I worked with a lot of the turtles and servers, things like that. Yeah.
You said that the stony coral disease popped up around 2014. At the same time, on the West Coast, we saw sea star wasting disease pop up around 2014. It coincided with an anomalous heat wave, ocean heat wave. Was there anything like that? Anything that was analogous to that popping up around the same time?
Not that I know of. I don't have enough expertise to speak to that. other than there's a, there's a sea urchin issue that's going on in the Caribbean right now. There's various issues, and how they're connected, I don't quite know, but there may be connections there. I remember when I lived in Alaska. So I lived in Juneau, Alaska, on Hawk Bay, and that was, we could watch the sea star wasting disease and the impacts that it had there. But the actual connections, I don't know, but your point's well taken. That maybe there's other things that are happening there. Yes.
Bruce, can I call you Dr. J?
Yeah, you should.
Really cool.
You've never seen me play. My dad went to, he was an excellent basketball player, but he did not pass at all. So,
You probably don't know this off the top of your head, but the two deep sea corals that look morphologically very distinct. You know what genetic loci they looked at?
that I'm not positive. Again, as a manager, most of the time I just know the results at the end, but I should know. Hopefully this is being recorded, right? I wish I knew more. I could find out more, Thomas.
It's not a big deal, just.
I'm not sure exactly what those slides. We have done things like, so as Alaska, we worked on different, like what species they are. So we worked on sand lamps that had that came from Japan. And we used there some of the mitochondrial markers to show that they were how diverse they were. So I'm not sure if they were mitochondrial markers. That's generally what I use and work with people with that.
So we got time for one more question. We have one? Yes, please.
Hi, maybe it's not completely science related, but as we young scientists, sometimes we try to do something for oceans and corals and everything, and we face frustration many times. By your experience, how do you manage that type of frustration and keep going to propose solutions instead of just sitting and crying?
That's just a tremendously insightful question along the way too, because I experienced the same frustrations that everybody feels at different times along the way. It's just the patience necessary to get through some of the things that you have to go through. I have seen things switch in pendulum, so I've been around long enough to watch one way versus another. Again, it might get me a little bit of trouble, but I try to anticipate. Sometimes you'll have somebody behind you, right? And they'll say, I need you to go left. Because they're responding to whatever pressures they have. They need to go right or left or this or what they do something. And when I first started, I would try to do that because you want to do the best job and people have an insight of what you want to do. They're like, well, you need to go in this direction. And you have to take a shift to try to move it in that direction. And it takes a while to move it and get into that direction. And by the time I got over there, they had moved. They said, no, we need to go back over there. And then I could never catch up. I was always behind. So I try to understand when I go through with, the people that I work with, not necessarily all the things they're telling me to do, but understand underneath it, what's driving it. And try to anticipate what are the needs going to be, not necessarily right now, but what are the needs going to be in 2 years from now? So the organization, the group that I have is best positioned to answer the questions that they really need. So some of it's in a way. But great question. I too get frustrated. But doing the nerdo with the students, so it's just to say thank you. You brought a lot to me too. I know it was great working with you all. Hopefully that worked out well. It's hard to do it virtually, but to me it helped a lot too. Like I never used Chime before. We both use Chime and on mainline language and things like this. So I got just as much out of it as everything else. So thank you.